So I'm sure, like most of you, I'm really interested in the diversity of organisms that we see in the world. For example, all these species within the sunflower genus Helianthus. And in particular, I'm interested in why this diversity is not contiguous, or why it's patchy. And so, as I'm sure you guys all guessed, I'm here to tell you a little bit about my work on speciation. One of the classic ways that people study speciation is by measuring reproductive barriers or traits that stop gene flow between groups. And the idea is that if we understand how these barriers evolved, then we're going to understand better how different species become reproductively isolated. When you're doing this type of study, it's especially informative to look at groups that are really early in that divergence, and that's because it will allow you to see which barriers actually drove the species apart, rather than those that accumulated after divergence was complete, or speciation was complete. So, I'm looking, for my PhD, I wanted to do exactly that type of study. So what I set out to do was measure reproductive barriers between incipient species within the species Helianthus pedularis. The typical ecotype in this species sort of occupies sandy soils across the central US. You see them driving across the road all over the central US. The other ecotype lives in these really impressive sand dunes at Great Sand Dunes National Park in Colorado. If you get the chance, it's worth visiting. And throughout the talk, whenever I refer to the sand sheet ecotype, those will be in blue, the dune ecotype will be in red, and any hybrids will be shown in purple. And the other really nice thing about this system is that we know from the glacial history, as well as from data from microsats and rad sequencing, that the divergence is probably less than 10,000 years old. So we have a pretty good idea that we are looking at early in the speciation process. Okay, so I said I wanted to measure reproductive barriers, and I managed to measure 7. Those were selection against immigrants, flowering time differences, differences in the pollinator communities, pollen competition, F1 hybrid germination, that's supposed to be a seed, not an eggplant, and hybrid pollen viability, and finally selection against hybrids. So I could talk for this whole time about the methods that we used to measure these barriers, and I've done that before, so today I'm going to skip straight to the results, but if you're interested in the methods, please come talk to me about them later. And the results are that some of the barriers were strong. And the barriers that we found, I think, are pretty consistent with what we would have expected in what we think is a case of ecological speciation with gene flow. So we found strong selection against immigrants, differences in the pollinator communities, selection against hybrids, and perhaps the one surprising thing that we found was strong pollen competition. Okay, so this was really satisfying. Great, we found barriers, awesome, that makes sense. But we wanted to take this a little bit further and look a little bit deeper into a few of the barriers, in particular trying to get at the genetic architecture of some of the traits that might be underlying them, as well as trying to take a guess at the forces that might have actually caused these barriers to rise. One of the reasons we were particularly interested in genetic architecture is that my colleague Rose Andrew did some great work that showed that there are a few large regions of genomic divergence between the species. So these are regions that have significantly higher FST. And we expect that the traits underlying reproductive barriers will map to these regions. And we were also curious about whether multiple barriers would map to the same regions, which is something you might expect in a case of speciation with gene flow. Okay, so let's start with selection against immigrants and hybrids. It's relatively easy to understand how these barriers might have arisen. If you have divergent natural selection in 2 different environments, that will often lead to local adaptation, which is essentially selection against immigrants. Furthermore, if the hybrids have an intermediate phenotype that don't match either of the environments, you can get selection against those hybrids, which is the other barrier that we found. And in this case, it's great because we think we actually know, we have a trait that we think is under divergent selection in the 2 environments that might be causing this, and that is seed size. So dune seeds in the system are about 3 times heavier than sand sheet seeds. Here I'm showing 1,000 seeds from one dune plant and 1,000 seeds from one sand sheet plant, so the difference is pretty noticeable. Furthermore, data from reciprocal transplants in which we put both the pure ecotypes and the hybrids into the sand sheet environment and into the dune environment suggests that seed size is under selection. In both environments, heavier seeds emerge better. Here you can see this is an estimate of emergence rate and the various seed weights across the bottom. And this makes sense, especially in a system where there's loose sand. You can imagine that if you are a sunflower seedling and you get buried in a bunch of sand, that's not going to be great for you. So if you have a big seed, hopefully you can get up and out of the sand pretty quickly. On the other hand, we found that sand sheet plants make way more seeds on the sand sheet than in any of the other plant or environment combinations. And we think that's at least partly to do with the fact that they can make many, many, many small seeds. So overall, we think that there's a sort of trade-off-like process here between seed size and number, where dune plants do better on the dunes because they make large seeds that emerge better, and sand sheet plants do better on the sand sheet because they make many small seeds. So overall, I think we have a pretty good hypothesis about the forces that might have led to selection against immigrants and hybrids, and we have a trait that we think is worth trying to QTL map. Let's go back and talk about pollen competition for a minute. So this is a reproductive barrier that arises when you put equal amounts of pollen from both ecotypes. So in this case, I made mixes of dune and sand sheet pollen, and we put those on multiple plants. In this graph, each line represents a single mom, essentially. Red's dune, blue is sand sheet plants. And then on the Y, you can see what I'm graphing is the proportion of offspring that are sired by dune plants. So anything that has a line that doesn't overlap zero, overlap this 50% line here, those are plants that are significantly biased towards siring seeds with dune fathers, and anything that is below here, those are plants that are significantly biased towards seeds that are sired by sand sheet fathers. And you can see that overall, dune moms prefer dune dads, and sand sheet moms prefer sand sheet dads, but there's a lot of variation in whether or not siring is actually biased, or what we're calling choosy in this case. Okay, so we didn't really have a great hypothesis as to why choosiness arose in the system. So what we wanted to do was also map this trait itself, so choosiness, in addition to the seed size trait we talked about earlier. And the way that we did that is we made F2 mapping populations for seed size, and we sequenced using a reduced representation library method, in this case GBS, to sequence the top and bottom 5% of the distribution. And then we also sequenced all the plants that we had already phenotyped for choosiness. In the end, we found 2 QTL for seed size, and these overlap with the regions of divergence that were found earlier, which is great. That suggests that seed size really is likely to underlie a reproductive barrier and therefore be helping genetic differentiation. And we found that choosiness maps to the exact same area on linkage group 11 as that seed size trait. That's also really interesting because it suggests that it's possible that choosiness may have arisen through a pleiotropic trait, or that basically tight genetic linkage to a trait under selection facilitated the evolution of choosiness in the system. We're now exploring both the possibilities of pleiotropy and linkage. We have a little bit of data, preliminary data, that suggests that linkage might be slightly more likely. And again, I don't have time to go through those data right now, but I would love to talk about it after this if you want to hear about it. So overall, I would say that we are definitely starting to get a good grasp of the barriers that are separated, keeping dune and sand sheet sunflowers separate in the system, and that it seems like both natural selection and genetic architecture were important to that process. With that, I would like to thank you and everybody that helped make this work possible, and I'm happy to take questions.